Hello everyone. So I'm Maxime Hebrard. I'm from the Genome Institute of Singapore and from Precision Health Research Singapore. So working here today, we'll talk to you about the whole genome sequencing quality control standard. And so this effort started with a very simple observation that I think we can all agree is there is an increasing number of whole genome sequencing that are produced worldwide, and it would be extremely valuable if we were able to combine those different data sets, and we would empower research and create a real impact on the community if we could cross-analyze those different data sets. But there is a few different problems when we are trying to combine data sets that are produced in different countries by different institutes, and each project has its different focus. And so I will kind of focus my talk today on one specific problem, that is there is a lack of uniformity in methods to produce data, and especially how to assess the quality of the data. And so a simple example of how do you compute the depth coverage of one sample, and do you look at the full genome? Do you exclude chromosome X and Y? Do you look at only mappable region? What are you doing with duplicate reads? Do you count the duplicate reads or not when you compute your average depth? All those parameters impact a lot the final measurement of the depth coverage. And so there is a lack of uniformity. There is valid reason to count the duplicate reads or not, and different labs, different data producer measures the same metric different way. And so we went, with my colleague, we actually engaged with the Global Alliance for Genomic and Health, the GIW, that is a non-for-profit organization that have representation at the international level, and that is setting up a guideline and standard for sharing genomic and health data. And so this organization actually try to set guideline and standard all the way through the life cycle of a data. So from the guideline for consent, your patient guideline, and a file format for sequencings. They are maintaining BAM, they are maintaining VCF formats. There is guideline for data access committee. What do you look at? And there is a bunch of API and standard for exchanging data. And that's kind of, we went to actually engage with this group to say, okay, we have a problem of how the international community could set a standard for whole genome sequencing quality control. And so we had our first work group meeting, so under the umbrella of G4GH, in June 21, with a few of international representation from Canada, Europe, and Asia with us at Singapore. We draft our roadmap in 22. We work on our different deliverable. I will come back into that in few slides. In January 2023, so at the beginning of this year, we actually become official G4GH products. So now we are our official standard of G4GH that is in development. And in September, so last month, we went at the G4GH 11 plenary meeting, and we present this work to the different national initiative and precision medicine initiative, and get a lot of feedback that was very interesting. So what we have produce and what is available as the result of this working group as of today, it's we have focused our work on having a formal definition of 9 QC metrics that is currently focused on the alignment of short reads of human whole genome sequencing, because we need to start the discussion somewhere, so we need to focus a bit on our work. So we are focusing on the alignment of short read sequencing, and we have a list of 9 metrics with a formal definition in plain English, with every term is well defined, and the community agree of that definition. We have information about implementation. So we describe how we have one implementation to compute these metrics. And we are also suggesting some different tools and assessing if the different tools are functionally equivalent in to compute the same metric and respect the same definition. So our first deliverable is a formal definition of the metric on which everyone agrees. One important point is the working group, we are not telling people your data is good and your data is bad. We are not setting thresholds, because the thresholds depend on what you want to do with the data. What we are focusing on in the description of the metric and the method to compute it, to be able to compare a cat to a cat. So the second product that we actually share with the community is a reference implementation of a Nextflow pipeline that is available on GitHub, that with a set of tools that are packaged in the Docker image. And this pipeline is registered and available on Dockstore. So Dockstore is a platform widely used by GA4GH, where you can actually search for a pipeline, and in few clicks you can actually deploy the pipeline and run it on your data. And so that makes this pipeline searchable and easily usable by the community. And so the output of this pipeline, it's JSON files that you see in the corner of the slide with the sample ID and the list of the metrics and the value of the metrics. Another tool that we provide is actually benchmark resources, where we have a list of 100 BAM from the 1000 Genome project, so it's publicly available data. Those samples have been reprocessed with Dragon. The data is available, and we use this 100 BAM as benchmark resources. And so we are running our pipeline on those 100 BAM. We are providing a Jupyter notebook, simple notebook that allow to visualize the data. And we are welcoming other initiatives to actually implement their own pipeline to compute their own metric. And these benchmark resources can be used to actually compare the results and see if the different implementation is functionally equivalent or not. As a first application, we actually run this pipeline on the first 10,000 WG samples that we produce at Singapore as part of the National Precision Medicine Program. And it cost us about 25 cents per sample. So the pipeline handle BAM or CRAM. For this program, it's CRAM, so eventually the cost for BAM might differ. And I mean, that was a first run at that scale for the pipeline. So we are we are looking at some improvement for that pipeline. So don't be too scared by the cost. It's not fully optimized yet. It was just the first round, but it works. And so we have the data. You see the visualization and the distribution of the different metric on that slide. So what's next for our program is we are working on assessing different tools and looking at the functionality equivalence, because eventually we have made some choice, but someone can argue that there is another tool that computes the same metric, and we need to assess if it's equivalent result or not. We are obviously planning to extend our list of QC, and we are looking toward adding variant calling QC, so based on VCFs. We are in the middle of the discussion with a group to agree on the definition and draft the formal definition first, then the implementation will come. And we are also working very closely with GA4GH to integrate this work with the other standard of the community. And one thing that we are very looking toward is to add this list of QC as part of the data discovery. That would say, before you even download the data, you can already review those QC and see if that data set pass your own threshold of quality, because eventually, if it don't pass your own threshold, you don't need to put the effort to request access, download the data, and things like that. And here's my last slide to thank you all the people that are part of the discussion and the work group in our initiative at GA4GH. If you are interested and your logo is not on that slide, please contact us. We have a monthly meeting by Zoom. It's at 7 p.m. It's at Singapore, depending on which country you are. It can vary the whole day. And the last meeting, my slide is not updated, is the 28th of November.
All right, thanks Maxime for this wonderful talk, and also thanks to GA4GH for establishing all these QC standards. So any questions, please approach to the mic stand. Right, so personally I have one question for you. So we know that the QC tools are like omnipresent in every bioinformatics pipelines. So actually computational optimization is actually very important. It deserves a lot of efforts. So here I want to know more about what's your opinion on planning for these kinds of optimization for QC tools.
Yeah, so optimization it's always a discussion. As you saw, we were developing our pipeline of 100 samples. It works quite well, and then when we scale up to 10,000, we have some new problems. Mm-hmm. So we are working on that. There is also kind of, we are very split in the team, because there is the effort at GA4GH. GA4GH is focused on developing standard, and so eventually what is important for GA4GH is the formal definition and the fact that it's a consensus in the community. GA4GH pushes for reference implementation, like we need to have a tool that computes those. Now, we had some discussion. The fact that the tool is efficient is outside of the scope of GA4GH. It's like, you need to show you have a tool, it's working, but the efficiency is not really a focus on GA4GH. Now, I also have few hats, and as part of the national initiatives, then we have actual data. We QC actual data, and then the efficiency become important, because we don't want to spend hours of days to do compute. And here, things that I saw from few years ago to current, it's Go clouds, highly parallelized compute. and that solves most of the problem.
Yes, so my understanding is like it is like a iterative process that you need to like first
Yes.
to have something and then get something done and then you to have like, receive more requests.
Yes, so we start with 100, we went for 10,000, we will increase the number and see how it scales. Usually if it passes the 10,000, it's scalable for a few more thousand from my experience.
Yeah,
there is a first problem at around 5,000, and then if you reach 10,000, usually you still have more room.
Yeah.
And so
yeah, that makes
but
sense.
scaling is definitely, you need to have the data, test it, and improve.
Yeah. Okay. Let's have questions from the audience.
Yeah, hi Maxime, great work, very important. So I'm part of GA4GH, so I understand exactly how important and how labor intensive all the night calls have been to get to some kind of standards. But I just wanted to ask, obviously in San Francisco, you said you had a lot of different feedback. I'm actually quite curious about that feedback because obviously the standards, there's one level of standards for research, and then there's another level of standards for clinical, and then the divide between the two. So I'm curious, when you presented this, as you did mention when you talked about that during that it was interesting the feedback you had. I wonder whether you want to share, is the standards going to be adopted by the clinical end, like clinical laboratories that they're going to say that if they do this, this is the minimum standards, etc. So because as we all know in the community, commercial labs right now that's offering this as a testing, it's very, very variable standards at the moment. There's no clinical grade standardization. But I know GA4GH is much more on the research front than it is on the clinical. I wondered whether if you want to share any of that. Yeah.
So for now the discussion is led by researchers. So we haven't got much feedback about the clinical side of it. But I do agree that that's something important. And also, again, two different hats. For the GA4GH standards, for these standards, we are looking at create a standard for the metric definition and create a standard of how to compute it. We are not putting a standard into what is good or bad.
I see.
So for the clinical labs, what you were saying is like, they would be interesting, I hope, to comply to the matrix definition and comply to how it's computed. But then the threshold of what passed or failed the clinical lab or the clinical certification, for now is not a focus on the group, I would say. The threshold is not a focus, but we can talk definitely.
All right, we have room for one more question.
Okay, I just want to clarify one detail. Can we use all of these standards for non-human data? For example, I work with rodents, my colleagues work with plants or insects. Can we use all of these standards for our data, or we need to kind of adjust?
So I guess, so currently, most of the members of the group and the discussion are interested into humans.
Yeah.
Now, it would be very interesting to actually kind of test it and see what is relevant or not in the definition of the metric, what is relevant or not for plants or for other organisms, because I would expect that the main definition will stick. Now you might have other consideration, like additional consideration for plants, for example. And that would be very interesting to discuss and see how much we can align on that.
Okay, thank you very much.
Thanks.
Alvin, let us hear your question. Yeah.
Okay, no, it's really nice to see your work for GA4GH because I think at the end of the day, large consortiums need to prepare the field for the future as well. So I'm actually thinking whether, like, in your discussions, have you sort of thought about any implementations in like federated approaches, or to look at essentially with GDPR, for example, sharing will be totally different in like 5 to 10 years. Has that been an issue in GA4GH, for example?
So I would say, so for that group of QC standard, we are just looking at the QC standard. In the grand umbrella of GA4GH, there is a lot of discussion about, okay, what is needed to share data across country, and it's a headache. It's a lot of discussion, a lot of effort that come both from the kind of the lawyer arm of GA4GH and also from the developer, like to try to find technical solutions. Personally, I think the technical solution is not here yet.
Okay.
But I mean, I know that there is very skillful people working on that. So we will get there eventually.
Thank you.